Hola, 8th grade. Welcome to your lab for this week. Today we're going to be looking at how scientists work with tiny molecules that they cannot see. This is what we call gel electrophoresis. And I wanted to give you basically how this relates to us in modern day life. So if you're ever interested in how detectives and forensic scientists figure out if a criminal is indeed responsible for a crime, they usually use this method to measure the DNA that was collected at the crime against the criminal's DNA. If the sizes match, then more than likely the criminal committed the crime. So that is why we use gel electrophoresis. I'm going to read through the lab just in case you're not able to use Flash on your computer, and also help you out with your exit ticket. So it behooves you to listen. Okay, let's get started. You're holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? If the DNA strands are as big as your shoelaces, you could sort them by hand into groups and measure them. But DNA strands are molecules so tiny that you can't see them even under most microscopes. Is there a way to sort and measure the DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How does this work? So again, like I explained to you, this is particularly used in forensic science so that they can match the DNA found at the crime to the DNA of the suspected criminal. They don't match, criminal didn't commit the crime. They match, high chances that the criminal committed the crime. The gel is a filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it. So what you're seeing there is a zoomed in version of the gel. And basically, I want you to think of the gel like a sponge, like that you use to wash something. The holes are pretty even and spread throughout the entire sponge, or the gel in this case. And so that is how the DNA is able to move throughout the gel. We place DNA samples into holes at one end of the gel. So the gel or our sponge has these specific holes at the very end of it, and that is where you squeeze your DNA liquid or the samples into them. And basically, once we start running electrical current through it, they're going to move from these holes or these wells to the other side of the gel or our sponge, whatever you want to call it. Electrophoresis is how we push the DNA strands through the gel filter. By adding an electrical current, we can make the DNA move. So if you look at this diagram here, you see negative pushes the DNA down to the positive, and we're going to learn about why that happens. Short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. So again, once we add electrical current, the DNA starts to move through the gel, and basically the shorter ones will move furthest away from the well, and the longer ones will be closest to the well. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. So the reason why they show up looking like these small rectangles is because, like I said in the beginning, the gel has these holes spread throughout it evenly, and so that's why they all kind of look the same. However, you're going to notice this one's longer than this one, and their shapes are different because the DNA strand lengths are different. Now it's your turn to run a gel. That's scientist slang for gel electrophoresis. Follow along with the steps shown above. So let's look at our procedure. Step 1, make the gel. Step 2, set up the gel apparatus. Step 3, load the DNA sample into the gel. Step 4, hook up the electrical current and run the gel. Step 5, stain the gel and analyze the results. Here's what you'll need to make a gel: powdered agarose, buffer, a flask, a microwave, the gel mold, and the gel cone. Making an electrophoresis gel is a lot like making jello, but you don't want to eat this type of gel. Put a small amount of agarose into the flask. Agarose is a dried powder similar to gelatin, but made from seaweed. So we know gelatin is basically made from pig. So this is made from plant-based. Add some liquid buffer to the flask. The buffer is a saltwater solution that will let electrical charges flow through the gel. We've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. Open the microwave by pressing the open button. Place the flask containing the buffer and agarose mixture inside the microwave. Heat the mixture until the agarose melts into the buffer. Microwave, put it here. We've removed the plastic wrap from the top of the flask. Pour the melted agarose mixture into the mold. Notice that the mold has tape on each end to hold in the melted agarose. So we'll put here. Place the comb into the gel on one end. The notches in the gel mold hold it in place. So now we're going to make our wells by using this comb. Let the gel cool and solidify. This usually takes about half an hour, but we'll speed up the clock for you. As the gel cools, tiny holes will form in it. Carefully remove the comb, leaving empty wells for the DNA samples. Your gel is now ready to run. So now that it's solidified, remove the comb. Now it's time to set up the electrophoresis box. You will need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. Place the gel, still in the mold, into the electrophoresis box. We have removed the tape from the end of the gel mold. The gel should be just barely submerged in the buffer. The buffer conducts the electrical current from one end of the gel to the other. It will also keep the gel from drying out during the experiment. We are now ready to load the DNA sample into the gel. Here's what you'll need: loading buffer, the tube of DNA, the DNA size standard, a micropipetter, the electrophoresis box containing buffer and the gel, and pipette tips. Now again, we're creating this gel so that we can measure the size of DNA strands and compare them. With a clean pipette tip, use a micropipetter to suck up some loading buffer, then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load it directly into a well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy. This makes the DNA sample thicker so that it will drop into the well instead of floating away. The DNA size standard already contains loading buffer. Next, you will use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. Next, oops, my bad. Eject the DNA sample from the pipette into the first well of the gel. Nice job. In real life, loading the sample into the wells takes some practice, so don't be disappointed if you miss your target the first few times. Using a clean pipette tip, use the micropipetter to suck up some DNA size standard. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the lengths of the DNA strands in your sample. So now let's take it back to our example of solving a crime. This DNA size standard is the DNA sample from the crime scene. This DNA sample is our DNA sample from our criminal. So if this measures against this, we found our criminal. If it doesn't, then we didn't find our criminal. Oh, sorry. It's time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. The red end will generate a positive charge. Together, they will pass the current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. Now, the reason for that is because if you know about charges, you know that opposite charges attract and same charges or similar charges repel. So if DNA already has a negative charge, you want to put the negative charge or the black cord near to the DNA because they're going to repel each other. Negatives don't want to be near each other. So the DNA is going to do its best to move away from the negative charge. And then it's going to go towards the positive one because opposites attract. So that's why we put negative closest to the DNA and positive towards the opposite end. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location on the power supply where you want to plug in the black cord. So click here. That's right. Now click the location on the power supply where you want to plug in the red cord. Turn on the power supply. Your gel is off and running. Let's look into the gel box to see what's happening. Check for any tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof that a current is running. So here's your negative cord, and on the opposite side there is your positive cord. Repelled by the negative charge, DNA moves through the gel toward the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than long strands. Over time, the shorter DNA strands will migrate farther from the starting point than the longer strands. We can't actually see the migrating DNA bands, but we can see the blue dye from the loading buffer as it migrates. So as you can see, once we turned it on, that DNA was out of those wells and moving. You've finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using DNA staining solution. Again, remember, we cannot see DNA with the naked eye, so therefore staining it will make it visible. This stain is a chemical called ethidium bromide, which binds to DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. Drag the gel out of the mold and put it into the DNA staining solution. Because ethidium bromide binds to DNA, it can damage the DNA in your cells. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. So basically what it's saying is you should never handle this without gloves because the solution is strong enough to destroy your own cells. It takes about half an hour to stain the DNA in the gel. We'll speed up the clock for you. Remove the gel from the staining solution and place it on the UV or ultraviolet light box. So we use ultraviolet light. Turn on the UV box. Ultraviolet light from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. Alrighty then, here's our results. Now you can determine the approximate lengths of the DNA strands in your sample. Compare the bands from the DNA sample with the bands of the known length from the DNA size standard. Enter the estimated length in base pairs, because DNA is a bunch of base pairs, so that's why base pairs are its units. For each band in your DNA sample. Electrophoresis cannot tell us the exact DNA lengths of DNA strands, just a good estimate. So give it your best guess based on the DNA size standards. And I'll press forward when I'm finished to check my answers. So let's look here. Here's our sample and here's the standard. This one is lined up right against this one. So this one is about 6,000 base pairs long. Okay. Let's look at this one. This one comes in between. So not necessarily lined up with anyone. It's in the middle. So between 3,000 and 4,000. So I'm going to estimate it to be 3,500 base pairs. And then this last one, again, not lined up with any of the ones that we have here. It's in the middle between 1,000 and 2,000. So I'm going to estimate it to be 1,500 base pairs. Now let's check my work. Bam, all of my estimations were correct. The estimated length in base pairs of each band in your DNA sample is based on how they line up with the bands of the known length in the DNA size standard. The top band is approximately 6,000 base pairs, the second is approximately 3,500 base pairs, and the third is approximately 1,500 base pairs. Congratulations, you have just run your own DNA gel electrophoresis experiment. So now go and do your exit ticket and do well.